Okay. Thank you so much for having me. And it's actually great to be speaking after Nikolai again on non-human subjects. I'm currently a researcher in Professor Omri Koren's lab in Bar-Ilan University in Israel, where most of my research is on humans, focusing on the microbiome in the first 1,000 days of life. But now I'd like to share with you a pet project of mine, a continuation from my PhD research, and a little bit of ecology and conservation. So why is microbiome important in ecology and conservation? Just like in human microbiome studies, in animal studies, we can understand a lot about the overall host state, breeding success, and pathogen load from the microbiome. We can define a core microbiome of functionality and examine different core dynamics based on seasonal changes and also environmental changes over the life history, whether it's migration, mating, or some other activity. And of course, we can profile and compare between different populations within the same species or similar species. And this is really important in the realm of conservation, where we do captive breeding and reintroduction into the wild. And there's been some studies showing that across species, the microbiomes of captive and wild animals differ. And one of the ways that could potentially improve reintroductions could be through microbiome manipulation. This leads us to the question of how best to sample microbiomes in wild free-ranging species. And of course, in ecology and conservation, it's best to do as little damage as you can. And also, many of the animals we work with can't easily be trapped. So in a lot of population genetic studies or breeding strategy studies of relatedness and kinship, non-invasive samples of shed feathers, hair, fecal samples are used to extract DNA. And similar to what we're doing now with COVID-19, we can assess animal pathogens or disease states like avian flu through environmental sampling like water, fecal sampling, and carcasses. So this brought me to the question, why aren't we doing this in ecological studies and conservation studies? Why aren't we using non-invasive sampling? And so here I want to show you a little proof of concept, POC, of how we can non-invasively sample birds, and we compared it to birds that we actually did trap to see if it's even any good. And after that, I'll dive into to a study we performed using this non-invasive method, so you can see how some cool real-life results. So first, what did we do? This study is on the common crane, or the Eurasian crane. In Latin, it's called Grus. And all of the fieldwork was done as part of Sasha's PhD. We couldn't have done it without her. And so there are two arms of this study. First was the group of trapped cranes. And because this is a very sensitive animal to trapping, when it's exposed to high levels of stress, it greatly diminishes survival, either with immediate mortality events or delayed mortality. These birds are trapped with sedatives, with a food bait laced with oral sedatives. And then after the birds fall asleep, they're collected and they're brought to a handling area. And as they wake up, they defecate, and samples are collected from the ground from these defecations. And this is considered the trapped group. In parallel, we observed birds in nearby fields that defecated naturally, and we approached them, and then once they flew away, we sampled these fresh droppings. And then we used 16S RNA sequencing to characterize the microbiota. From a first glance, you can see that the trapped samples have much richer diversity, even at the phylum level, which is actually a really good thing for us because we're very nervous about getting an increase of contaminants or aerobic bacterial increases in the non-invasive samples, and that's not something we see off the bat. However, when we examine the beta diversity of our two groups, in yellow you have these birds that were trapped, and in orange the birds that were not trapped or the non-invasive samples, we see a significant separation. We also see increased dispersion in our trapped group, which matches our phylum bar plot. And this leads us to conclude that even if we don't have contaminants in our samples, something is going on, and we have two distinctly characterized microbial communities. We dug a bit deeper to examine differentially represented taxa. In general, 60% of the genera sampled using these two methods were shared between the methods, and only 13 taxa were unique to the non-invasive sampling. So again, it doesn't seem like we have an increase of contamination going on. And I forgot to mention, but everything was processed with internal controls in the lab, and all of that was filtered out using decontam. So we're talking about contaminants in the field. Looking specifically at the differentially abundant taxa, we don't see a super clear pattern of what's going on here. And interestingly, less than a third of the differentially abundant taxa were prevalent in the majority of birds. Along the same lines, the relative abundance of these taxa was also very, very low, with only 4% having greater than 0.1% of abundance. So together, this suggests that the differentially abundant taxa may be minority taxa. We also looked at the functionality of the taxa that were differentially represented, and luckily we didn't find any soil or aerobic contaminants. We examined taxa that might be associated with stress as a result of trapping, and these also were differentially abundant. But something else that we had to consider was the immediate effect of the oral sedative on our birds. And here we did find an effect. One of the differentially abundant families has been associated in the past with oral sedative use. So to sum up our proof of concept, that's this first section, different sampling methods do lead to different microbiota, but when considering what this means, it might be okay if we're trying to identify a core microbiota or a core across time. The majority of taxa were conserved, though fine-scale profiling might be difficult using one or both of these methods. Experimental validation can definitely help. If we have birds in captivity, we can collect fresh samples and delayed sampling. And we can also examine the effects of immediate effects of controlled stressing or sedative use. In general, working in the field can induce stress in animals. Here we didn't see the effect. We did see the effect of sedatives, and this raises the question of maybe non-invasive sampling is even better than trapping. And here I'm going to show you a study where we could only sample non-invasively, and we think our results are reliable based on what I've showed you until now. So in this study, we again are tracking the common crane, and we're interested in seeing how seasonal changes and diet shifts affect its microbiome. And there's a specific conservation punch here I'll tell you in just a minute. So again, this is the common crane. I'm currently in Israel, and in Israel, in the north of Israel, we have a place called the Hula Valley, which is on the main migration pathway. And in the winter, we have hundreds and thousands of cranes, which is really, really cool. But of course, having such a high concentration of these birds can lead to conflicts with farmers. And so we have an artificial feeding station where we feed the cranes with corn, almost solely with corn, during the majority of the wintering in a certain area of this farmscape, such that they won't forage in the fields of the farmers, and they are maintained in this feeding area. And in this study, we integrated movement data with GPS tags that we affixed to these birds with non-invasive fecal samples. And in just a minute, you'll see why we had to use non-invasive fecal samples. So here's a lot all at once, but this is the first thing I wanted you to see. We tagged all of our birds in Russia. And here, technically, we could have sampled these birds, and we did for microbiome profiling. But later on, we didn't retrap these birds. We just have GPS tracks. And in Israel, in the wintering sites, we can't trap the birds. It's very, very challenging. So in Israel, we had to use non-invasive sampling techniques, collections from the fields. Which this is what we also did in Russia to make sure that all of our samples were comparable. So here we trapped and tagged with GPS trackers 30 birds. And in parallel, we also sampled an additional 30 birds. There's thousands of birds in Russia, and the likelihood that a tagged bird was sampled is very slim. So it's important to keep in mind that these are parallel data sets, they're not completely overlapping. When examining, this is the actual movement data, we see that we have an early autumn sample. These are the birds in Russia before they migrated. A late autumn sample, the birds arriving in Israel immediately after the migration. A winter in Israel sample, which you can see around here in blue. These are birds that wintered in Israel but did not use the feeding station. And in red in the north, we have this feeding station site. And interestingly, the birds in the feeding station spend almost all of their foraging in the feeding station, maybe a little bit outside of it here. But these wintering birds in Israel that didn't use the feeding station, even if they visited the area, here you can see them, they never foraged here. So we have pretty separate diet behaviors of the birds that we sampled. So the question is, what does this mean for the microbiome? And we actually found that our differences tracked. We have some seasonal differences In green and in yellow, we have the 2 autumn samples, and they're not significantly different. And I actually think this is really surprising because one sampling in yellow is from Russia before the migration, and the second is from Israel following migration. So not only have these birds undergone a very challenging migration, but they've also been exposed to slightly different diets. Of course, in the winter, the diets shift, the crops shift, and there's a change. And the birds that have been at the feeding station have the biggest shift in microbiome, suggesting that maybe feeding isn't great. So again, food supplementation, diet, and season hierarchically affected the microbiome composition, but we think non-invasive sampling is actually a really great method, especially in this type of study design. We have other co-results. You can read the paper. You'll see a barcode in a second. You can email me. We did differential abundance analyses and all of that. But my take-home message here is that you can really do great things in the field, in ecology, without disrupting our species. And I think this is just a good example of that. So thank you so much for listening to the Movement Ecology Lab, where I did my PhD, and the Kren Lab, where I'm currently working, and to all of you for listening.